Okay, so for today, what we want to get through in about 30 minutes is just pinpointing some things from Chapter 9, from 9.1 and 9.2 on DNA replication and mutation. There are a number of really great videos that I have saved for you, and I'm just going to open that up here and just show you, remind you about these videos.
Where was the extra credit? Because I don't have that in my.
It's there now. I didn't get to it yesterday. I'm going to confess I took my son hiking, which was an hour and a half of driving each way just to do a 5-mile hike. But I had to get away from the computer for a while. So it's up now, and the due date is June 6th because it'll make them, you're doing a DNA lab next week. And so it makes the most sense for you to have the lab done and to have the extra credit be due next week after. Okay.
Yeah, I'm excited to hear you teach about the DNA stuff, but honestly, the lab is bugging me. I don't know if it's the way the questions are written, but I just.
Oh, this week's lab on fermentation?
Yeah.
Okay. Well, let's save that for the end. Okay.
Okay.
So we'll do DNA, and then I want to talk a little bit about mitosis. And then since I didn't get a pre-recorded Zoom up, I'll probably make a pre-recorded Zoom up for mitosis. And then I need to, oh, I know what I want to do. I'm just going to put these two side by side here, if it'll let me. There we go. So I have side by side Canvas tabs open. So the one on the left shows you the videos for DNA, and the one on the right, I'm going to open the content objectives for this week. And the first one says, summarize the experimental design and key role of each of the following: Watson and Crick, Rosalind Franklin, Griffith, Hershey and Chase, Avery, McCarthy, and McLeod, and Chargaff. A couple of these guys and women are referred to in my PowerPoint, but more specifically, to understand the full history, it's this history of DNA, the Bozeman Science one on the top left, okay? And then the second bullet point, explain why DNA in one cell contains the blueprint for every cell in a eukaryote. That's covered, you know, when in the Bozeman Science, where he talks about the history of DNA and where he talks about DNA replication. And then I'm going to pop into the PowerPoint in a minute and explain to you where that is answered. And then the third bullet point about the structure of the DNA. This Bozeman Science DNA replication, the Amoeba Sisters DNA replication, those are all going to be really helpful for that. And there's also some great information in this PowerPoint. And then if you were to go back to the biological molecules PowerPoint, there's some preliminary information you got on nucleic acids there as well. Then this next one that says, outline the general features of prokaryotic and eukaryotic chromosomes, including their packaging. I'm going to talk about that briefly in the PowerPoint. And also the Amoeba Sisters and the Bozeman Science would go through that. So like if I click this one open.
How do you do the split screen?
I have a split screen right now. Yeah, and I know it's making it small for you, but you could open, you know, I'm just showing you as I go through. So like this is point number 4 in the checklist.
No, I meant how would I do it? I can't figure out how to do it.
Just a second, I want that muted. The way that I did it, so like if you have multiple tabs open, if you watch this, if I click on it, I left click and I pull, it pops it out. So now it's its own window, not just a tab, and then I can slide it around. And in fact, if I push it back up there, oh, that wasn't what I meant to do. There is a way to just pop it back in.
Cool.
And see, I popped it back into my row of tabs. Okay.
On yours, both of them fit. On mine, they don't both fit at the same time. They kind of
Well,
cover each other.
The reason mine are fitting is because I'm using a second monitor and I'm
Oh,
doing a screen extension.
okay.
I do a lot of grading. I have a 15-inch screen laptop, or maybe it's only 14. It's a pretty good size, but when I split screen there, it's relatively small. And I do have, you know, not too young and not too old of eyes and pretty decent eyesight. And I've had lots of years of practice of zooming in and out, grading online. But for you and to be able to show you things, I have this second monitor so I can pull things up and make them bigger. So you're welcome. This one on DNA replication that's running right now, Paul's going through a little bit of the cell cycle just to put it into context and explain how DNA is replicated before cells can divide. And the point of the video right now is he's talking about the original ideas that people had about how DNA got copied and how it got from original cells to the cells that that cell divided into. And so he's talking about semi-conservative, conservative, and dispersive. And this is really useful. If I show you on the 6th bullet point over here on the right, it says describe the process of DNA replication. And it talks about the semi-conservative nature. So watching this video would be really helpful to understand the history and how people thought DNA was replicated and how they tested it. And then basically how they disproved things. And then I've zoomed forward and how they actually figured out that DNA replication was semi-conservative, meaning that the original strands get split open down the middle, and then enzymes add new nucleotide bases opposite the bases, the pre-existing bases in the split open strands, so that at the end, each strand is made of an old half and a new half. And so I'm just kind of skipping forward where he's talking about anti-parallel right now. So that's really important. That's covered here. And then he's going to show you steps of DNA replication and all the enzymes that are involved. And that's really, really important also, because it's got to be very, very well monitored and protected. Otherwise, every time DNA is copied, you'd end up with a problem in the daughter cell, and then maybe you'd have DNA coding for proteins that didn't function properly. And then what was the point of investing all this time and energy into copying the DNA to go into the new cells? So this 7th bullet point here says being familiar with the sequence of replication and the following components. So this video walks through all of them, and I'm going to tell you this video that I found really, really does an amazing job of going through in detail these 2 bullet points, and it is worth every single one of your 40 minutes. It's going to be like I was lecturing to you, and I'm going to show that. I'm not going to play it right now. I'm going to pop it up here and put it into I'm going to show you. It's 40 minutes, but if I back up here, he starts with showing you 5' to 3', showing you the phosphate and the sugar and the nitrogenous base, this green thing here. And he labels everything, and he talks about 5' and 3'. And, you know, basically he's giving my lecture for me. Then he explains semi-conservative, and he draws it, okay? And I'm skipping ahead. He shows you the blue is the old strands, and the green is the newly copied strands that are complementary to the old, okay? And then he goes through and he explains how this process happens. what each enzyme is and what it does. And so then he explains how in eukaryotic DNA, because it's double-stranded like this, and because it's linear, as in it's not circular, so it has two open ends, he explains how in multiple places along each linear strand that there's an enzyme that it's going to break hydrogen bonds and it's going to open the strands, and that at each point where he's drawing that green dashed line is an origin of replication, a place where the DNA was opened and where enzymes can now do the work of synthesizing the new strand of DNA that's going to join to each of these original strands. And so he draws them getting bigger and explains how the enzymes do that. And I'm just kind of fast forwarding you through how, see the red, this is our newly copied DNA. And so he explains how as time goes on, the replication bubbles grow, and then eventually what's going to happen to them? They will merge, they'll run into each other. And so you'll have original parent DNA that's blue, and then what's he drawing here? He's drawing the red daughter DNA that was newly synthesized that now becomes an individual strand. And then what happens once these guys separate? Then we have two semi-conservative strands that are completed, and one has an original strand of DNA, the blue, and each one has a newly copied strand, the red. And now we have this chromosome has been copied, and it's ready for the next step in the life of the cell, which is for the nucleus to be split, and then for the cytoplasm to be split, so that then the daughter cells each have the same DNA amount and the same chromosomes coded for as the original. And so that's why we're covering DNA replication in chapter 9 before we cover mitosis in chapter 6, because you can't divide the cell correctly until you copy the DNA that's going to go into those two cells. Okay, so he goes through all of that, which is about 10 minutes in. Okay, and then he's going to tell you, so how does that process actually happen? And now he's going to draw you one replication bubble and zoom in. So like I said, with all the marker colors, he's doing exactly what I would have done on the board for you. And it's worth every minute. Even if you watch it 3 times, it's worth 2 hours of your time.
Okay.
I'm serious. It totally is. Okay, so now do you see how he's zoomed in really big and he's got this opened parental DNA, the 2 strands of the double helix, and he shows the point where it gets open is called the origin of replication, and that's his dashed line. And so he's going to walk you right through. See, I've skipped ahead a little bit. Come on, let me have it. And he's labeled and he's showing you the direction. And 5' to 3' has to do with the sugar and where the carbons are. And it has to do with the carbons in the 5-carbon sugar and where the 5th carbon is pointing. And on each of these strands, the carbons don't point in the same direction. And so the strands are anti-parallel. The carbon molecules point in opposite directions. And why do we care? The reason we care is the enzyme whose job it is to add new bases can only grab on to this 3' OH or hydroxyl group at the end of a 3' carbon that's hanging out there. And so DNA is synthesized in the 5' to 3' direction because of the limitation of the enzyme that's doing the reaction, okay? So he's done the labeling for you, and I'm going to skip ahead a little bit to now he starts to fill in. I'm just waiting a little bit here for my computer to catch up. He starts to fill in the enzymes and show you and talk about the direction, and he explains why it's 3' to 5' or why it's 5' to 3', and he goes through the direction of synthesis And then he basically explains why, therefore, you have continuous or ongoing replication toward the replication fork on one side. And then once he gets out of the way, he'll actually show you how there is a discontinuous or fragmented synthesis on the other half of the replication bubble on this side, because you can only synthesize 5' to 3'. And it's taken a while to get there. So see how he drew these little fragments? It's because he's showing you how that is going to occur. And in the extra credit assignment, I put in a couple of videos, and one of them is a simulation, like a digitized simulation that shows DNA being copied and shows the leading and the lagging strand. And the other one I think is just a link to an Amoeba Sisters video. So I'm gonna skip ahead here, and you can see he's drawing really fast right now. And we get you towards the end, and can you see how he's got this whole thing all labeled? And he explains how each half of the replication bubble is just an upside down. Do you see how this one is labeled leading on the top left, and this one is labeled leading on the bottom right? Mm-hmm. The old strand here is running 3' to 5', but this one is being synthesized in the opposite direction. So this is 5, and this is toward the 3'. And so this can be continuous, 5 to 3', but because the replication bubble only opens a little bit at a time, as it opens and the enzyme can get in there and start the work, it makes a little section of new DNA. And then the bubble has opened more, and another new little section is made, and then another and another and another, and so on. Until we join up with another bubble over here. And then there's another enzyme whose job it is to seal all of these new DNA fragments together, okay? So for your extra credit assignment, you're not drawing this on the board, but watching this will give you an understanding of all the components. And what you are doing is finding household objects like string and rubber bands and pennies and paper clips, etc., to represent the different parts of the replication bubble. And then you're going to record a little video of you explaining how DNA is replicated and explaining what the different components represent. So if you had a button that was going to slide along and smooth out the coiled double helix, that's called topoisomerase, you would show in your video you moving the little button and you doing that. And we do this in lab when we have lab together, but we don't. So I'm giving you the opportunity to just earn a little extra credit by practicing it on your own, okay? So if I move to the end, he just finishes his labeling and his explanation, and it's just an amazing video as far as getting all of the pieces in there. For your lab next week, one of the things that you'll be doing, and that I've always put on the lab practical, do you see how this is just a computer image of the replication bubble and all the different components? Well, do you know what ends up on the lab test? Is one where these are all blank and you have to label them all. On a take-home test, it won't be the same. It'll be more like you matching up the role with the location, so what is it that the RNA primer does? And so that, there's a lot of vocab here.
Yeah.
And that's part of the reason why we're splitting it into 2 weeks. But that is this thing. Be familiar with the sequence of replication and the following components. And I list those terms, and it says what are these structures and what is their function. So that's why it's, wait to watch that video I just showed you, or I'm going to pop the link up in here in the videos, until after you have watched several of these other shorter ones, okay? And then go in and watch that. The history of DNA video and the Bozeman science and the DNA Amoeba Sisters replication, those will explain the central dogma of DNA to RNA and protein, and so does the PowerPoint. And then these two right here, DNA nucleotides and how they can be mutated, that's these last two videos that are Bozeman Science and Amoeba Sisters, okay? And then you get into mitosis with a whole bunch more lovely terminology. And if I go back here to the module, I curated a bunch more videos under chapter 6, and I'm just scrolling down here. And it's these right here, where all of these, where the cell cycle is explained, where mutation and cancer are tied together. And so you'll see, if you watch 3 or 4 of these, you'll see them all pulled together again, okay? Excuse me. So how about if I pull open the PowerPoint now that I had going on in the background here. And I just want to quickly, so I'm going to take about 15 minutes right now and walk you through some key things. I'm not going to spend a bunch of time on the overall history that you need to answer in your notes because it's in the video. What I want to zoom in on is Rosalind Franklin was a scientist working independently in Maurice Wilkins' lab. And she was using this crystallography technique where you actually freeze proteins to study the structure of protein. And then she started using the same frozen X-ray imaging technique to study the structure of DNA. And people had been studying DNA and figuring out that DNA carried the genetic material for, you know, a couple of decades, but we were still trying to identify the actual structure of DNA and how it was put together. They didn't know sugar phosphate, nucleotide, beyond that they knew those were the components. They were trying to figure out how they all fit together. So this image that's here next to her picture is a really famous picture in biology because it was one of the pictures she took where do you see these the dark smear on the top and the dark smear on the bottom? Those are actually the sugar, the 5-carbon sugars and the phosphates that are stacked on top of each other in the outside. And if you were to like take a rubber band and attach things to the inside of it and then twist it, the outside structure of the rubber band would create these dark patterns like what you see here. And then inside, you see these little lines that make an X? Those are actually the 4 nucleotide bases that are hydrogen bonded down the center. And she hadn't figured out exactly what it was. She was studying it. Well, do you know what Wilkins did? He gave James Watson and Francis Crick, who were also studying the structure of DNA, a copy of her picture. He didn't ask her, he just gave it to them. And they were able to use it successfully to create a 3-dimensional model of DNA. And that's the basis for the Nobel Prize that they won. And she actually died before they won the Nobel Prize. And currently you can't get the Nobel Prize posthumously, but back then you could. But she wasn't considered. And it's basically because her work got, you know, borrowed without a permission. So it's just kind of an interesting comment on, you know, progress and competition in science can be pretty fierce, and it wasn't exactly ethical what happened, but
that is not how the Bozeman video described it.
I mean, they had, Watson and Crick had a lot of information. They probably would have figured it out eventually.
Yeah.
And it's not like they stole it.
Yeah.
But it was given to them. And
yeah.
So she didn't get credit for her contribution. But if you watch that history video, you can see that we didn't just jump to the end with Watson and Crick and 3-dimensional structure. it took, you know, dozens of scientists studying DNA to figure out that DNA was indeed the biological molecule that carries genetic or hereditary information, and how it's composed of, you know, phosphate and 5-carbon sugars and nitrogenous bases, and how they're hydrogen bonded together, how it's a double helix, how it's anti-parallel, etc. People once thought that proteins may have been the genetic material, so we had to disprove that idea. This next slide is the one that answers the next bullet point in the content objectives. That basically, you know, why is DNA a blueprint? Because DNA is a molecule that codes for your genes, and your genes code for your proteins. And what do your proteins do? Well, what is this second point? Proteins do plus or minus everything for the cell. Proteins do all the work in the cell. And so, I mean, it should say in, but if proteins do all the work, then the DNA codes for everything that a cell builds, everything that a cell does. And so DNA is basically the blueprint for everything that makes a cell alive. So without DNA, there would be no functioning cell. And
What
all
number
of
is
your,
that?
that's
What slide is
3.
that?
Slide 3.
Thank you.
Here, I can make this just a little bit bigger. So basically, we figured out, you know, through all the history that you're going to go through, that DNA contains the genetic information. Well, what does that mean? Whether you have 46 chromosomes in a human cell, or, you know, 3 times that in a shark cell, in a given individual cell, that cell has all of the chromosomes. They're not all being expressed. They're not all being turned into proteins. Only certain ones are basically turned on and turned off. That's genetic expression. That's why you have neurons and muscle cells and lung cells, etc., because certain genes are on and certain genes are off, determining what proteins are made for that cell and what its structure is going to be. But the DNA in every single cell contains all the chromosomes and therefore all the genes. So you could technically, if you knew how, take a cell from any part of the human body and you could clone that human if we had the technology. Because what's contained in that cell? The blueprint of every cell, you know, all the genes for that individual. I'm not saying it would be ethical, just that if we had the technology, we could do it, okay? And then if you think about it, why are we studying embryos so extensively? It's because those cells aren't yet specialized. And so a lot more of those genes are still turned on as opposed to turned off. And if we could figure out how to direct cells that are dividing to become specialized tissues that we needed, then we could replace damaged tissue. And if we could figure out how to do it without needing embryos, I would be much more excited and on board, but that's just my personal ethical philosophy about that. I'm not trying to sway you one way or the other. And the reason I would want to be able to develop specialized tissues is my oldest daughter was born blind, and she has what's called optic nerve hypoplasia. And during the 7th or 8th week of pregnancy is when the optic nerve tissue divides and specializes, and she just doesn't have full optic nerves. And so she doesn't, her eyes are fine and her brain is okay, but the connection is bad. And wouldn't it be great if we could figure out how to implant tissue that would then grow optic nerves? On the printer downstairs. Shut the door, please. Thank you. All right, so when we look at this next slide, it just says, hey, in a eukaryote, there's so much DNA that where is it kept? It's kept it in the nucleus. It's kept it, wow, that was great. Stored in the nucleus behind a membrane, where in a prokaryote, it's not. So, and because DNA stays in the nucleus, how do we make proteins? How do we get those genes expressed? We have to have an intermediate. We have to have RNA carry a temporary copy of a length of the DNA that's going to then be read and converted into proteins. So the second half of chapter 9 is about how DNA is read and turned into RNA, transcription, and then how RNA is read and turned into proteins, translation. But for now, we're focusing on replication and mutation. So this just summarizes that overall process of how the genetic code is used to make proteins. And so DNA is transcribed to RNA and RNA is translated into protein. Those are terms, transcription and translation. Those are the nouns, and transcribe is the verb and translate is the verb. It's really important for you to understand that because DNA itself isn't doing the work, DNA is the code for the proteins that are going to do the work, and we have to make a temporary readable copy of sections of DNA that code for proteins, and that is what a gene is, a length of DNA that codes for a protein. This next diagram you've seen before, that just turns DNA to RNA to protein into a picture, if it helps you to remember a little bit better. Then in the next slide, this is slide 7, there's a review from chapter 2 where we went over nucleic acid structure that shows you that DNA and RNA both consist of 3 main parts. A 5-carbon sugar, ribose for RNA, deoxyribose for DNA, a phosphate, which should be a little bit familiar to you with the P in the middle and the oxygens around the side, and then what we call a nitrogenous base, which is a ring structure, which may be single ring or double ring. And why do we call it a base? I'm thinking about just the way that it behaves, but more that it's nitrogenous because in the ring is not just carbon, but also nitrogen. You can see in these rings down here that the carbons are labeled, but it's not just carbon, there are some nitrogen in there as well. And cytosine, guanine, and adenine are 3 of the 5 bases, and those are found in both DNA and RNA. But we find thymine in DNA only and uracil in RNA only. And so when you practice for your learning assignments this week and for your lab assignment next week, you'll be practicing DNA replication and transcription and translation. You'll be practicing using letters C, G, T, A, and U for the nucleotide bases that are, the thing that holds the code. So what holds the code in DNA and RNA? It's the sequence of those bases. And why do we care? Because a 3-base sequence codes for an amino acid. And then the sequence of amino acids codes for a protein, as you learned in chapter 3. Okay, so how's the information used in a cell? But remember, DNA is kept in the nucleus in a eukaryote, and the RNA is single-stranded. It has ribose and it has uracil, and then basically it takes that temporary copy of the DNA and it's used to make protein. Okay, this slide shows you those 4 bases with their symbols, C, G, A, and T for DNA, C, G, A, and U for ribose, and then it shows you the difference in the sugar. They're both 5-carbon sugars, but it's deoxyribose for DNA and ribose for RNA. The only difference between the 2 sugars, and if I come back here to slide 7, do you see up here in red OH right there, and then H right there? There's no oxygen attached to the 2nd carbon for deoxyribose. So D means without, deoxy without oxygen. Deoxyribose is the same as ribose, but without the oxygen. And it just makes it a little bit different. This slide shows you that the yellow, see the yellow stripes here? It's a model and it's meant to be three-dimensional, but obviously on a computer, everything is 2D. These yellow stripes are the phosphates and the blue stripes are the sugars. So what's the sequence in the helix? Phosphate, sugar, phosphate, sugar, phosphate, sugar. You get the idea? And what sticks out into the middle? The nitrogenous bases, okay? And the two strands run, they coil next to each other, but then the nitrogenous bases stick out towards each other. And these little red dots that you see on the left-hand side here and between the two strands, those are hydrogen bonds. So individually, a single hydrogen bond is weak and it's temporary, right? But these hydrogen bonds between the nitrogenous bases, say between like 1 million base pairs, how strong do you think that would be?
Pretty strong.
Pretty strong, especially internally protected by the sugar phosphate backbone. It's so strong that we don't rip the DNA apart very readily, and it takes an enzyme to uncoil the DNA and smooth it out, and then another enzyme to go through and snip all those hydrogen bonds so that we can separate the strands and copy those nitrogenous bases. Is there a question?
Yeah, I was watching some of the videos yesterday, and I'm trying to remember if there was something about how you don't want the individual strands to touch each other, so the hydrogen bonds keep them apart as well.
Yeah, that would work. Also,
Okay.
if DNA gets, and your DNA gets broken in many cells many times a day, and the cell usually does a really great job of fixing it.
Okay.
So, but if the strands get smushed together and then they both break and you have a double strand break, replacing DNA where it's been busted straight through is very hard to do correctly, because then you have no template strand and you're missing part of the DNA. And so the cell will sometimes try to correct that, but oftentimes that's a signal that will send it into apoptosis, programmed cell death.
Okay.
Which means the cell's just going to get recycled, all the components. Okay, the next slide shows you the base pairing. So when we look at DNA to DNA, it's A to T, T to A, C to G, G to C. But when we're going to copy DNA, opposite the C in DNA, the G will go in RNA. Opposite the G in DNA, the C will go in RNA. So that's identical to copying the DNA. But opposite the T in DNA would put an A if we're showing what's going to be in the RNA strand. But opposite an A, there will be a U. We're not putting a T in there, we're putting a U because RNA has uracil, not thymine. And then the reason we care about base pairing between different RNAs is there's more than just messenger RNA, the temporary copy of the DNA. There's also tRNA, which is transfer RNA, and there's rRNA, ribosomal RNA. And those base pair C to G, G to C, U to A, and A to U. See how there's no T in there? And we're going to learn about base pairing between mRNA and tRNA next week. This is just a heads up that it's coming, okay? Chapter 9, by the way, it's all in one PowerPoint, but I think it's only to slide 20 that's relevant for this week, okay?
Oh, that's good to know.
Okay?
Up to slide
So
20.
we'll get there in a second and I'll confirm that. So this is a reminder of the base pairing, and it gives you a visual. When you're copying DNA, what goes opposite A? U. What goes opposite C? G. What goes opposite T? A. What goes opposite C? The G. And in copying DNA and when matching it up, if you're going to build a strand, what would go opposite the A? T. Opposite C? G. Opposite T? A. And opposite C? G. See how it's always C and G together and T and A together? Whether it's A to T or T to A, they base pair. You never find C with A, or T with G, or T with C, or A with G. And this is what Chargaff was able to figure out, the base pairing, which was part of what led to Watson and Crick to be able to develop their three-dimensional model that showed the base pairing accurately in the center. If I back up here, remember there are single rings and double rings. Which ones do you think stick out further?
Double.
The double, right? Now look, here's T as a single and A as a double. Oh, so if they pair up in the middle, they're going to push the rungs of the ladder, the sugar phosphate chains, a certain distance apart. What if A and G paired together? Two double rings. They would like push it out further. And what if C and T paired together? There would be a pinch in. And so instead of being smooth, uniform, diameter, the double helix would pinch in and out, in and out, in and out. And it would be very, I don't know, it would be like driving on the pothole dirt road the last 2 miles to the trailhead yesterday. And it would be easier for it to catch on things and to break. So it makes sense that through evolution, we get base pairing of A to T, T to A, and C to G and G to C, so that the cell has the ability to monitor base pairing and to make sure that the 2 strands of the double helix are kept equidistant throughout the length of the double helix chromosome. Okay, so now we're going to go, slide 12 walks you through packaging that prokaryotes tend to have one single circular chromosome and not linear, so it doesn't have ends, and it's not behind a nuclear membrane, but it is kind of contained by the cytoskeleton in a region of the cytoplasm we label as the nucleoid. And since DNA is a working molecule, it doesn't just like get put in a safe deposit box and never looked at. It's in a protected place, not in permanent storage. And that means it has to be read to produce molecules like proteins, and therefore it has to be accessible. And so it may be in the nucleoid region in a prokaryote or in the nucleus in a eukaryote, but it still has to be accessible. And it's only when the DNA, it's only when the cell is going to divide that the DNA gets tightly packed into chromosomes. Otherwise, it's more loosely coiled into what we call chromatin, and it's wrapped around these special proteins that we call histones, and it forms this kind of ball and string model where it's loosely coiled around these histone proteins to form the subunit structure of proteins called the nucleosome. And it basically just loosely stores the DNA to keep it from getting damaged and to make sure that it doesn't get broken really easy. If you do any boating, when you take the boat off the dock, you don't just like throw the rope all over. You coil it up carefully so that you can use it when you need it and so it doesn't become a tangled mess, right? Well, that's what the cell does with protein. So here on this slide, you see there's this DNA double helix where the light blue is the sugar phosphate backbone. And inside these colored sticks are the nucleotides that are hydrogen bonded to each other. But imagine that like a single chromosome could be hundreds of feet long. So how does it stay inside the nucleus of a cell? Well, it has to be coiled around a protein. See these little alpha helices in here? This is a single coil around a protein called a histone. And basically we call that a nucleosome, and there are multiple nucleosomes that then also coil themselves. And so we have some levels of coiling that exist inside the cell at all times so that the DNA is protected from damage and so that it can fit nicely. How much can you fit in a suitcase when you fold things neatly or roll them or even use like vacuum sealing packaging, right? A lot. How much can you fit in the suitcase if you just throw all your dirty laundry in there? Not nearly so much, right? And so, yeah, so the nucleus, I'm thinking about all the laundry that I haven't done for the last 5 days. The nucleus isn't very big and cells are tiny, but they have to hold a lot of DNA. So we have to keep it somewhat packaged except for when it's being used, okay? So there is a couple of levels of coiling. The subunit, oh, excuse me, wrong way. The subunit of DNA is the nucleosome, the DNA wrapped around the histone protein. And then those chains of DNA around the histones, those nucleosomes, make extra loops. And so we have, this level of packaging at all times except for when a length of DNA is being read. And then when we want to divide the cell, we condense the chromatin even further on itself and then tightly pack it into a chromosome, okay? So this slide, number 14, one of the learning objectives says to compare and contrast prokaryotes and eukaryotes. This slide, in addition to the video talking about prokaryotes and eukaryotes, would be really helpful because prokaryotes are circular DNA. They only open to replicate in one spot. Eukaryotes have multiple origins of replication because they have to be able to replicate along the whole length of both strands of the DNA. You can see that prokaryotes have much faster replication, 1,000 nucleotides per second compared to 50 to 100. So they're like 10 to 20 times faster, prokaryotes are. They have less DNA overall in the cell, and because it's circular DNA, it just starts on one point and it's continuous, where eukaryotes have continuous replication on one side of a bubble and discontinuous on the other, and so it just takes longer. Telomerase is an enzyme that exists basically to protect to protect the linear ends of eukaryotic DNA from getting chopped and getting shorter and shorter and shorter every single time DNA gets replicated and a cell divides. And in fact, as you age, part of the reason that you age is that your telomeres, your little protective ends on your DNA, you don't lose your actual DNA that codes for genes, but your telomeres, the protective caps or on the ends of your linear DNA, they get shorter over time. And it's actually one of the hallmarks of Alzheimer's is shortened telomeres. Okay, so let's see. I said, yeah, slide. Okay, so 21 is the one that there's a YouTube link that explains the importance of telomeres. That's right there in the slide. Okay, and then 15, 16, and 17 go through semi-conservative. And after you watch the video, looking at these slides will review for you the semi-conservative model that basically what do we get in the daughter cell? The two daughter cells, the two cells that are made from the original, we have half of the DNA is original parent DNA and half is newly synthesized DNA. And so semi means partial and conservative means is it conserved or retained? And we conserve part of the original DNA in each of the daughter cells. It's not that this original DNA goes to one and then the other one is all new. And it's not that it gets all chopped up and then swapped all over the place. It's that there's, in the new double helix, one of the strands is complete and the original parent strand, and the other half of that double strand is newly synthesized, and that happens for both sides. Okay. This one is just to remind you, hey, what do you need to learn? All of the what? Yeah, all of the different components.
So is this still part of this week?
Yeah,
learning?
That's still part of this week. Then 19 and 20 are vocabulary terms that define these guys. So after you've watched a couple of videos and you go back and you look at this or you draw it in your notes, then you've got the vocabulary here to go with it, okay? It's a reminder. The last, and I said 20 slides, it's actually 23. You want to go through slide 23. Yeah. Slide 23 is about mutation. There's a YouTube video link in here about mutation that is different from, and maybe I should just copy this and plop it in there too. This is, I'll show you. And I'll put it in at the bottom. But if I copy this guy in here, as long as the link is still good. It's loading. I'm muting it. And I have to push play and we have to get past the ad. Okay, so what this is going to show you is different ways in which DNA gets damaged. And then it's showing you, you know, hey, if it mismatches and we have to cut one out, and it shows how that happens. It goes through enzymes that repair breaks in the DNA, okay? And it goes through, oh, hey, if there's a missing segment, you know, how do we replace all of that? And then it even goes in and talks about like so how to cut out bases is one of the things I talked about. And then it says, what do we do if we want to replace a whole section? And then it even talks about what happens if both strands break. And so then it just shows you that. So it's a really nice animation, that was the term I was looking for, that walks you through different types of mutation and their impact on the DNA and how the cell tries to fix it. Your cell repairs your DNA, you know, like 1,000,000 times a day. Even though we have all these preventative mechanisms, when the DNA is copied, we're still repairing our DNA over and over and over again. So it sort of makes sense that in your lifetime, if you live long enough, you will eventually get cancer, which is you'll manage to have mutated DNA that gets copied into daughter cells that doesn't get destroyed. And then those cells kind of ignore cell cycle mechanisms that are supposed to control all of that. Okay, I need to make this small again. And then what I want to do here is talk about 14A and B, which are your DNA and mutation assignments. 14A is just walking you through the components here initially of the double helix and having you identify the parts of the sugar phosphate backbone and the base pairing in the center, okay? So that's model 1. Then it gives you a little bit of information about the history and the base pairing and you know, what was developed. And then model 2 shows you how the parent DNA is separated and how then the nucleotides get brought in and bonded together, okay? And it just walks you through briefly the steps of that process and asks you to explain semi-conservative and what that really means. And then here, this is page 3. So on page 3, there's a little bit about mutation and asks you to reflect on how semi-conservative replication might prevent mutations. Think about it. If there's always a sample, so if you want to make a cake, do you want to have the recipe book out the first 20 times you make that cake?
Yes.
Yeah. Well, what happens if there's no recipe book? Are you going to make the cake right?
Probably not.
Probably not. You know, if you're a baker and you've done it a few thousand times, oh yeah. But for a cell, having the parent strand still there is like having the cookbook out and you follow that parent strand and add nucleotide bases that are exactly opposite or complementary, and therefore you're less inclined to make mistakes in copying the DNA by following that base pairing. So semi-conservative replication provides complementary base pairing, and it means there's always a template, there's always something to copy, okay? So I have a question for you. If A always pairs with T, Are they going to be present in different amounts in a given cell or the same amount?
Same.
The same. So if adenine, if on a given length, if in a given cell, say humans, if there's 31 out of 100% is adenine, then there would be also 31% thymine, which would make 62 out of 100% of the nucleotide bases, leaving 38% to be cytosine and guanine. And 38 divided by 2 is 19 and 19. So what goes here for guanine? 19. And what goes here for thymine? 31. And what do they all add up to? 100. But the amount of adenine and thymine, guanine and cytosine is not the same in the cells of all eukaryotes. It varies. It's part of what enables us to have different genes coded for, because we don't have the same amounts of A and T and C and G, and the sequence of those bases vary. So why do we have different proteins in different organisms? Because the sequence of the DNA codes for different genes based on the sequence of the nucleotide or nitrogenous bases. Model 3 walks you through the cell cycle and makes you think about, hey, in S phase, synthesis, that's where DNA is replicated, which was partway through the regular life of the cell, where G1 and G2 refer to growth, and then M refers to mitosis, or when the nucleus divides. So you've just got to walk through and refer to the descriptions here and answer these couple of questions. And then you need to figure out which one of these represents semi-conservative replication, okay? And what would they look like? And remember from before. And then the last part is that first video that's in here. Let's see, where is it? The rest of this assignment just makes you watch a couple of the videos that are already listed, okay? That's what these are, and actually answer them, okay? So it's going to walk you through some of the vocabulary again. So that's 14A, and 14B is just walking you through some mutations.
14B.
Yeah, it's okay, because see, 2/3 of this page is taken up by the model, and there are 3 questions. And if you watch the video about mutation, it'll tell you what substitution, insertion, and deletion are. And in short, substitution is swapping one base for another. If an enzyme doesn't catch that there's a problem and you end up with a different nucleotide in there, then you've substituted a different nucleotide. You swapped one out. Insertion is adding an extra length inside, okay? And deletion is cutting at least one out. So in a nutshell, that's what substitution, insertion, and deletion are. So then there are some questions that walk you through practicing, you know, substitution and insertion and deletion, and you just have to look at how have these sequences changed. And then page 4 comes back and has you compare the results of the mutations. And I was considering having you wait to turn this one in until next week, because if you understand transcription and translation a little bit better, you might understand the consequences of mutation a little bit better. And I don't know if anybody would object if 14B got a due date that was kicked back till next week.
I forgot about 14B, so I'm cracking up.
Yeah, okay, so nobody would mind. Okay. because I put it in the order that I wanted you to do it, but so then that basically takes you through all of chapter 9 that you need, and chapter 6, that learning assignment there, you have the baby. I'm just going to pop open the learning assignment there for chapter 6. So this is the learning assignment that you're working on for chapter 6, and this walks you through the cell cycle, okay, and has you answer some questions about it, and then it explains to you how the movement of the cell through the parts of the cell cycle is affected by what's going on inside the cell and what's going on outside the cell. So extra would be outside, extracellular, and intra is inside the cell, okay? And what you really want to do before you try to answer this worksheet here, can you hear my daughter shouting?
Mm-hmm.
Is watch a couple of these videos on the cell cycle and mitosis, okay, before you try to answer this one. And then remember, if I go back here, I think it's buried underneath. Here are the content objectives that we were looking at before. When I make the video and I come back to this, you'll want to, it'll help you understand this terminology even better. And we'll have a lab that also has you practice it. That's your lab that's following the DNA lab. That's the next lab after that. And so watching these, I would read these through and then watch the videos and then come read them again. And remember that your study guide for chapter 6 isn't due until Monday, okay? So focus on getting chapter 9.1 and 9.2, these ones answered, and then start working through mitosis. And I will make another video that'll be like 30 minutes long that will help you with the cell cycle. And by video, I mean I'm going to do a drawing, not go through and answer all these questions for you. When you watch the videos here that are linked about chapter 6 and about the cell cycle, and you read about it and you go through the PowerPoint, it explains to you what G1, S, and G2 are.